And there's no weekly skills check next Monday, but there is a two-lecture activity. I think, do I read that right?
Goals for tonight.
Yes, there is goals for tonight. I started it last night. I was like, I'm going to do it again.
Yeah, there's no weekly skills check.
Have you looked at it yet?
You do need your eye clickers today. I'll put those out, please, and we have
I'm looking at this
lots to do.
Hey, baby.
Major pathways of fuel metabolism. You already know them all, so it's all three. What are your pathways? Glycolysis. And the opposing?
Gluconeogenesis.
Gluconeogenesis. What else?
Citric acid cycle.
Alright, you guys, citric acid cycle, sure. What else?
Glycogen synthesis and degradation.
Glycogen synthesis, degradation. Up at ETC with the citric acid cycle. Okay. What else?
Beta oxidation.
Okay, so we'll say fatty acid. We'll also say biosynthesis and degradation. And although, we briefly looked at that, we didn't much with that. And then the last one, amino acids. So we have protein, amino acid synthesis, and degradation, and again focusing mostly on the degradation side. Okay, so those are all some lovely pathways that we've done. All of these pathways are affected by the concentration of AMP. And so AMP actually is kind of a unifying signaling molecule for metabolic pathways. In some cases, it is a direct allosteric effector of metabolic enzymes. But probably the more important method, more importantly, it is an allosteric metabolic enzymes here. It's an allosteric effector of a kinase called AMP kinase. Which we haven't talked about yet. AMP promotes autophosphorylation of AMPK. So it phosphorylates itself. And then AMPK, when activated by phosphorylation, has lots of targets. And those include a functional enzyme, PFK2, FBPase2. It includes hormone-sensitive lipase, includes acetylcoenzyme A carboxylase. It includes glycogen synthasase. In general, as AMP increases, catabolic pathways are upregulated, they increase. Oh, sorry. And anabolic pathways go down. So there's all our pathways. Although the electron transport chain is just kind of shuffled down here somewhere after the citric acid cycle. So we've got protein, carbohydrate, and fat synthesis and degradation, and where they feed into glycolysis, the citric acid cycle. All right. So knowing what I just told you about AMP kinase, phosphorylation of PFK2 by AMPK in heart muscle will do what exactly? Most are saying increase. Not all of you are saying increase. Right. So PFK2, that is the thing that phosphorylates and makes my fructose bisphosphate, which was my allosteric regulator of PFK. And PFK is my glycolysis enzyme, and so we are going to increase that activity in the process. We're going to increase glycolysis. Decreased gluconeogenesis. Phosphorylation of acetylcoenzyme A carboxylase by AMP kinase. I have no idea what I meant to say there, but you get the idea. Will it increase or decrease the carboxylase activity? And so this is a question of where is that enzyme? What does it do? The name might tell you something. Okay, yeah, so we don't remember. That's okay. Acetylcoenzyme A carboxylase. If something's a carboxylase, what is it doing?
Biosynthesis.
It's in the name.
Taking away CO2?
Well, you have a choice. You can take away or you could add. If I take it away, I'm probably going to call it a D-carboxylase.
Okay.
So I'm adding, yeah. So I'm adding carbon dioxide to acetylcoenzyme A. Where are you going to see that? Why would I want to add a carbon to acetylcoenzyme A?
To make fatty acid.
To make a fatty acid. So this is all fatty acid synthesis stuff. Yeah. Which means we're going to decrease the activity because we're downregulating all our biosynthetic pathways. So that is your first committed step of fatty acid synthesis. And we're going to decrease that. All right, next one. Phosphorylation of hormone-sensitive lipase by AMK in adipose tissue will do what to its activity? saying increase. That would be right. Okay, so lipase is going to break down our stored fats, so that's moving things towards fatty acid oxidation and degradation. Okay, last one, phosphorylation of glycogen synthase by AMK in the liver due to what to its activity. You might remember glycogen synthase gets phosphorylated by a lot of different things. Glycogen synthase, what does glycogen synthase do?
Makes glycogen.
Makes glycogen. Is that going to be an anabolic or a catabolic pathway? An anabolic. So is AMK going to want to increase that or decrease that? Decrease. So it's going to decrease activity. Because we want to break things down, we want to make ATP. All of those. It is more key to kind of recognize what the enzymes do, and then remembering that when you're regulating, it's all going to be anabolic together, it's all going to be catabolic together. Metabolite flex in 5 tissues. There's a lot more in your book than we have time to talk about here. But your tissues are not all the same. They have different roles, and they all can't do the same metabolic processes. You mentioned a few of these before. So your brain. The brain uses about 20% of resting oxygen levels. Its primary fuel is what?
Glucose.
Equals glucose. Its backup fuel, if you're starving, is ketone bodies. The brain does not metabolize fats. It needs a constant fuel source. Your brain is very greedy. So normal blood glucose concentrations is going to be somewhere between 5 and 6 millimolar. If you are less than 5 millimolar, then you're going to experience this dysfunction. You're going to be groggy, you can't focus. Less than 2.5 millimolar, then at that point you're experiencing coma and eventually death. Even when your brain switches over to ketone bodies, you still need a certain percentage of glucose in the blood. So if you aren't eating it, your body has to figure out a way to make it. Okay, the other one that we kind of talked about a little bit is muscle. This is another greedy tissue. This uses about 30% of resting O2. Primary fuels includes just about everything. So you can use glucose, stored glycogen because it stores its own glycogen. It can do fatty acid metabolism, and it can use ketone bodies. Your muscle cannot do gluconeogenesis. So its glucose has to come from outside sources. And it cannot export glucose like your liver does. Okay, so even though it can store it as glycogen, just like your liver also stores glucose as glycogen, the liver can break it down and send it out. Your muscles cannot. When under exertion, your muscles are first going to use any existing ATP. So when your muscles are contracting, it's going to consume a lot of ATP. So that's the first source. The second source we talked about, maybe you remember.
Lactate?
Sorry?
Lactate?
That's coming later. We're going to talk about that later today.
Phosphocreatine.
Phosphocreatine. Yeah. Second is the phosphocreatine. So remember, ATP can be stored as phosphocreatine. That's something we talked about last semester, so that was a while ago. But that's only going to last you maybe 4 to 8 seconds. You're resisting ATP, you only have enough for maybe 1 or 2 seconds of work. It's gone. Okay. Third, is going to use glycolysis. So now you're looking at your glucose stores. But initially it's going to be anaerobic. How long will that last you?
10 seconds.
A few minutes, yeah, just like 1 or 2 minutes. And then you're going to kick into your aerobic use. So that is your CAC when you're going to ETC. Because the immediate need is ATP. You got to have the ATP to contract, and it's faster from the stores and then from anaerobic metabolism, but you can't continue on that. So yes, your sprinters are going to be able to do all anaerobic, but your long distance runners, it only takes a couple of minutes before they're switching over to a full citric acid cycle to get the ATP. Adipose tissue. We already talked about that. Stores what?
Fatty acid.
Fatty acids. And again, we're controlled by that hormone-sensitive lipase and glucose concentrations. Adipose tissue is boring. It is there to store fat. What's not boring is your liver, which is arguably the most important organ in your body. And so this maintains levels of metabolites throughout your body. Although it makes ketone bodies, it doesn't use them itself. Okay, so it makes ketone bodies to be able to send out to your brain or any other tissues that need it. But it won't use the ketone bodies itself. I mean, it's just going to break down the fats itselp to make ATPs, so it doesn't really need the ketone bodies. Under starvation conditions, it's going to convert fatty acids to ketone bodies. And it's probably going to rely on amino acids for its own fuel.
So then it doesn't use ketone bodies for fuel?
No, not at all.
So
It
why
makes
would
it for other tissues. So ketone bodies are export only.
Okay.
The isozyme of hexokinase in the liver is called glucokinase and has a broader range of activities at different glucose concentrations. Okay. And that's important because if you have glucose in the blood, if you're a tissue that needs glucose, it's just going to grab onto it and use it. It's going to take it, it's going to use it. But the liver needs to know when glucose can be imported to be stored and when glucose needs to be exported because other tissues need it. And so it has to have this wider range of activity over different concentrations of glucose. I have a figure to show you with that, but I think I want to write down our number 5 first before I do that. So the other one that's kind of important in here is the kidney. And this filters waste products from the blood. But it also scavenges metabolites for gluconeogenesis. And when you're under starving conditions, the kidney makes about 50% of your glucose. Okay, so like your liver, it can do gluconeogenesis.
Does it only do gluconeogenesis during starvation, or can it do it other times, just not as much?
Everything is at equilibrium and upregulation, downregulation. So sure, but it's not going to waste its energy, I guess, doing that if you don't need it. Okay, so who was it? Katie, you asked me whether you had to remember all the other stuff or not from the previous exam. Yeah. That is what you have to remember from the previous exam. So is this more of a where these things happen by tissue and what will be upregulated and downregulated in certain situations. It's all there. Okay. What we haven't talked about yet, which we're going to talk about next, is this business here with the breakdown of amino acids and the anaerobic respiration and how lactate is swapped between the liver and the muscle. So that's the one thing we have left to do.
Dr. O, how would you look at that?
To study it?
Yeah.
I probably wouldn't. That's the
Helpful.
show them and be amazed kind of figure. I don't find it as useful. So it's more useful for me to kind of calculate what are my tissues and what can they do and what can't they do and which fuels do they use. Okay.
I wish I would talk to my phone.
Do you really wish that same?
Yeah.
Oh, books and
Unless we could use it in
here's your
class.
glucokinase versus transcriptase.
$150
So hexokinase in other tissues, again, it's like turned on at your highest velocity, basically, as soon as glucose is around. But in the liver, it's not hyperbolic like that as much. It's not steep. Okay. We're making good time. I was ready to get through this. The Corey cycle. This is a metabolic cycle between the liver and the muscle. And it supports anaerobic exertion. So remember we talked about homolactic fermentation? And in that pyruvate got converted to what? Lactic acid, okay. So what happens is that your muscle sends lactate to the liver to be converted back into glucose via gluconeogenesis. And then the liver sends the glucose back to the muscle. We'll just say tissues because it doesn't necessarily have to go back to the muscle, but it's going to go back out there again. We're not going to talk about specific reactions here, partly because you already know them. We've already done them. We've already talked about the lactic acid fermentation. We've already talked about gluconeogenesis. So those pathways you already know. It's just the cycle that needs to be pointed out. And we will avoid talking about how lactate gets turned back into pyruvate. That's the one we're seeing here. So I'm in my muscle. The muscle, it can take glucose from the blood. It has its own glycogen stores. You break that down by glycolysis, and then if it's anaerobic, pyruvate gets turned into lactate. Lactate goes back into the bloodstream, and then it's picked up by the liver, and then it gets converted back into pyruvate and back glucose through gluconeogenesis, which gets shipped out again. So this is why it's a cycle, because they just trade back and forth under anaerobic conditions.
On that one it said glucose to glycogen. Is that just if there's enough glucose around? Otherwise it looks like it kind of skipped and went out again.
Yeah, no, it's the
So it doesn't have to go to glycogen
no, it
first.
doesn't have to go to
Okay.
glycogen. But if you stopped exerting, then it's going to pick up glucose and store it.
All right.
Okay, so that's one thing you should know. Another little pathway you should know for muscles is the glucose alanine shuttle or cycle. And this is an important one for starting conditions. So this pathway supports gluconeogenesis in the liver by breaking down proteins in the muscle. So this happens under starving conditions. This one we are going to draw just a little bit, although the reaction you've already seen. This is review. So any amino acid plus pyruvate which is an alpha keto acid. We haven't called it that before, but it is. Can be converted I'm having a good day here. To the alpha keto acid. Plus alanine. Because if you put an amino group on pyruvate, you get alanine. And the enzyme that does that is what did I do?
Transference.
It's a transferase. It's an amino
Amino.
transferase. I'm swapping an amino group for a keto group. Okay. That is what happens when we break down proteins for energy anyway, right? So the amino acids get converted to alpha keto acids. Alpha keto acids can feed into different places in the citric acid cycle. The amino group then, after it's on alpha ketoglutarate, that makes glutamate, then is oxidatively deaminated, and then we have ammonia. Okay. So that reaction you've seen, except instead of pyruvate, it was alpha ketoglutarate. We were looking at a different alpha keto acid. All right. So that's alanine. So in this cycle, the muscle converts pyruvate to alanine. Alanine is sent to the liver and is converted back into pyruvate. And that is then going to be used for gluconeogenesis. So I've taken a generic amino acid from my muscles. So I'm breaking down the proteins in my muscles and I'm going to get all 20 amino acids. I've taken the amino group and tacked it onto a product of glycolysis, which my muscles are going to be making anyway if they're breaking down glucose. And that alpha keto acid can then become possibly a citric acid cycle intermediate or a substrate for citric acid cycle. And the alanine that I've made then gets shipped out and goes to the liver, and then the liver is going to be able to convert that back into glucose. So it's a way to break down muscle tissue in order to keep your blood glucose levels high enough for your brain to live. So starvation diets, yes, you turn on all the signals to break down your fats. Yes, you also turn on all the signals to break down protein as well.
Do you know what the signal is?
Sorry?
What's the signal to break down protein?
Low glucose levels, in the muscle. No, I don't. And so I don't know if it's like a ubiquitin pathway that gets turned on or something has to get upregulated, obviously, and it's going to be key to a blood glucose level, but I'm not sure what that is.
Is it cortisol?
Mm-hmm. I don't know. Hormone controlled. Well, you see, if you're starving, then yeah, your catecholamines are going to get released, as well as your glucagon as well. So it could be a hormone control. It could be a substrate concentration of med school questions. Those are kinda, you'll know that better than me today. Okay, talking about hormonal control, that's kind of our last thing. I need to throw up the boxer diagram of the, as a Fourier cycle, there's our glucose alanine cycle. So, so the muscle breaking down the pyruvate, and then it transfers those amino groups, like alanine goes back here, pyruvate, glucose, and then the liver is what sends out ammonia and makes the urea. Okay, this is a kind of a full circle sort of thing. Hormonal control of metabolism, and again, this is pretty much entirely review. We've already done this. You've got some steroids and some peptides, and you have some catecholamines. And so we want to talk again just a little bit about insulin and glucagon. So insulin secretion comes from where? Pancreas. And is glucose levels that control secretion of insulin from the pancreas. Okay, and that's blood levels. And glucose can diffuse directly into pancreatic cells. So again, if the normal range of glucose is that kind of 5 to 6 millimolar, that doesn't do much of anything, but if insulin is released when levels start to get hot on the high side, so now we're going to be getting above 5, maybe above 6, we're going to secrete insulin to tell cells to kick that glucose up. The insulin response, that differs by tissue. Okay. Remember that the liver is always taking up and exporting, so it doesn't have to be told to take up glucose. If the glucose levels are high, it'll just take it up. But tissues, for example, muscles, use an insulin stimulated transport protein to bring in glucose. And all the glucose transporters are GLUTs, all caps. And then they have different numbers depending on what tissue you bring them in. Muscle is the major one's a GLUT4. And it is actually a very rapid response. And the way it can do this is by storing the transporters in vesicles inside the cell. So normally when there isn't high blood glucose around, there are these little vesicles inside the cell that have all these transporter proteins in them. And when insulin is around, insulin is going to bind an external receptor, and then part of the different pathways that get upregulated or downregulated, one such pathway triggers fusing of the vesicle with the cell surface. And so that increases the concentration of transporter proteins on the surface of the cell, and that will then increase transported glucose into the cell. And then later, when glucose is no longer around, then it brings them in the endocytosis and just brings them all back inside the cell, those vesicles. Okay, that way you don't have to synthesize the protein going all the way back to a genetic control, because that's slow. You want something really fast. So these things are always being made in high concentrations, but they're stored in vesicles inside the cell. Glucagon and catecholamines these again are opposite effects to insulin. Muscles lack glucagon receptors. There's no point in telling your muscles to do gluconeogenesis and send glucose out into the cells, so there's no point for them to have receptors. The liver has them. Yeah, I don't even know why I bothered to write this down. This is all review, so that's like random review stuff. Catecholamines, which are released with stress. And that includes low blood sugar. If you're hungry, that's stress. And these are also linked with adrenaline cyclase. So PK. The receptors, we talked way back, very first chapter of the semester. Insulin receptors were a kind of receptor. Protein, tyrosine kinases, or is there receptor, tyrosine kinases is the receptors. And glucagon, and your catecholamines, or G protein couple. Okay. So, these, you've already seen these. There's the insulin pathway. That's tons of stuff. But that includes uptake of glucose into cells. G protein coupled pathways, and this is the one PKA. So double cyclase and cyclic AMP, that's going to turn on PKA. And both glucagon and the catecholamines affect those pathways. That's my PIP2 pathway, and that's calcium levels. And remember, some of our pathways are controlled by calcium levels as well. Let's review. None of this is going to show up on the exam.
Thanks.
This kind of stuff might show up on the exam. So, what the tissue is, and what insulin or glucagon or epinephrine are going to tell them to do. Right? So, insulin in general is to take up glucose and then make glycogen. The liver, like I said, is more controlled about by glucose levels, so uptake of glucose is not affected, but glycogen synthesis is. Glucagon doesn't really affect the muscles, but epinephrine is going to tell your muscles that you need to break down your glycogen stores to gain the energy. So, this table is a good table. Ooh, okay. I'm going to show you the really fat mouse in a little bit, but what I really need to define. Yeah, kind of look like your hedgehog. I need to define diabetes for you. There are two major forms.
Diabetes.
First one is insulin dependent or juvenile onset. Or type 1 diabetes. And in this case, the pancreas lacks or has defective beta cells and doesn't produce enough insulin. Yeah. Okay, so type 1 diabetes is a lack of insulin. It can be treated with insulin injections. That can be tricky to monitor because if you take too much insulin and you don't have enough glucose in your bloodstream, you can do a self-induced coma or kill yourself because you basically OD'd on insulin. And so that's why if you are diabetic and you're doing insulin shots, you have to monitor your blood sugar. and the immediate danger is being too low in blood sugar. And so that's why you usually have some kind of sugary snack as an emergency piece of food. The other direction, okay, if you aren't taking insulin as much as you ought to and you're not keeping your blood sugar low enough, that is where you're going to start causing harm to different tissues. In particular, it shows up with your eyes. And so prolonged high blood sugar can usually cause blindness. And your kidneys is the other one that it harms your kidneys as well. So it can cause kidney failure. And the other type is insulin, or I would say non-insulin dependent, or adult onset, or type 2 diabetes. And in this case, the pancreas makes insulin. But the tissues don't respond to it. So sometimes we call this a insulin insensitivity. And it's not clear that every type of adult onset diabetes is actually the same cause. But it has something gets messed up between insulin binding a receptor and triggering export of the glucose transporter to take up the glucose. And so, where in that pathway it happens, that's not clear. And this one is not something you can treat with insulin shots because you already have insulin. That's not the problem. It has to be mostly monitored by diet. So you don't eat a lot of sugary food. And I'm out of time. Can I tell you about the fat mouse?
Yeah. I see it.
I didn't talk as much about diabetes this year as I have in previous years, and it had to do with my calendar, so it won't be much of the questions.
Oh my God.
Yeah, okay, so this is the obese mouse.
Just
This is a huge deal when it came out. So they found a gene that was linked to obesity in mice. And it was the OB gene, and so if you were homozygous for the recessive trait, then these mice would just eat meat, and they'd never stop eating. And so they called it the satiety gene, because there was some signal that was being turned off where they didn't know that they were full and they should stop eating. And it got linked to this little neuropeptide, so they got it all traced down to a neural signal. And then everybody was very excited because they thought, we're going to find the same neuropeptide in humans, which they did. Thought, we can just give them a pill and obesity will be cured. But then it didn't work that way. It never does, right? So, anyway, the picture is interesting to look at. But this is the OB gene codes for leptin, which is a little neuropeptide that sends signals from your brain to your stomach saying, stop eating, I'm full.
You're much cuter.
And doesn't really take something for us.
You look like Elsa without science.
If she were furry.
P, leptin. P is